Hello everyone. This is about the classification and nomenclature of the enzymes. Enzymes are biological catalysts that speed up the chemical reaction. According to the International Union of Biochemistry and Molecular Biology in the year 1955, they have been classified into many categories, and they have discovered the isolated and also studied different types of the enzymes based on the types of reactions. Each enzyme is designated like a unique 4-digit enzyme commission also number to identify its exact class, subclass, and also subclasses also they have been classified. Enzymes are basically classified into 6 classes, each with 4 to 13 subclasses and named according to their 4-digit number. So let's talk about the enzymes and their types also. The first one is considered as a oxidoreductases, enzyme commission also we can say 1. Function, it catalyzes oxidation as well as reduction reactions, that is transfer of electrons or hydrogen. For that, subclasses are dihydrogenases, reductases, oxidases, oxygenases, and peroxidases. Like A's and A's are the different types of the words being used. Substrate, which is reduced, and substrate which is going to be oxidized. They will be coming as a opposite version, that is substrate is oxidized and the substrate is reduced. Like this, the conformation will be decided as a oxidoreductases. Oxidation will be happening, at the same time reduction also will be happening. The key reagents heavily rely on this. Coenzymes like NAD, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, flavin adenine dinucleotide, flavin mononucleotide. These are very important for conversion of the NAD to NADH2 and NADH2 to NAD+. So like this, they will be having a energy constituent, especially in the photosynthesis and also respiration processes, and they act as electron carriers also. Example, reduction of this alcohol dehydrogenases, ethanol plus NAD plus, giving rise to acetaldehyde plus NADH2. So biological role is this specific reaction is vital for metabolizing alcohol in the human liver. Next, we'll talk about the transferases. Transfer of the group from between the substrate and S and S1, we call it as a transferases. So for example, substrate with G and substrate 1 is combining, and substrate are combined and G is being moved. So that means G is acting as a transferases, transferring the information. Transferases move a functional group from a donor molecule to an acceptor molecule. So the donor is usually high energy cofactors also we can call it. Subclasses like kinases transfer phosphate, transaminases transfer the amino group, and methyl transferases transfer the methyl group, and key reactants are ATP is the most common group of the donor in the kinase reactions. Hexokinases will be, and glucose plus ATP combined together form a glucose 6-phosphate plus ADP is giving rise to. Biological role, they are very important, critical and irreversible. First step of glycolysis, that is trans glucose inside the cell. So glucose is getting converting into glucose 6-phosphate is the first step in the glycolysis method during the respiration, which is common for the aerobic as well as the anaerobic reaction, which includes 10 steps. It is being followed by the other Krebs cycle and also ETS by aerobic processes. Otherwise, if it is anaerobic, it will undergo the fermentation process. Next, we see the hydrolases. The enzymes which catalyze the hydrolases of ester, ether, peptide, glycosidic, we call it as an hydrolases. Hydrolases breaks the covalent bonds by adding of water molecule, and they split a larger molecule into 2 smaller parts like esterases, peptidases, proteases, glycosidases, lipases, etc. Water acts as a nucleophile to attack the target bond also, peptide bond, ester bond, and glycosidic bond. For peptide bond, peptidases we need. For ester bond and esterases we need. And glycosidic bond, glycosidases. For lipid, lipases, like etc. And chymotrypsin, polypeptide plus H2O giving rise to shorter peptides also, secreted by the pancreas, and this enzyme is indispensable for digesting the dietary proteins in the small intestine. Next, lyases. Catalyze removal of the groups from the substrates by mechanism other than the hydrolases, leaving the bonds here. And here C X C Y, then X and Y plus C, like this. Lyases cleave this bond, carbon carbon bonding, carbon oxygen bonding, carbon nitrogen bonding, other bonds also by hydrolysis or oxidation method. This process usually generates a new double bond or a ring structure also. The reverse reaction, that is addition to double bonds, is catalyzed lyases also, decarboxylase, aldolases, dehydratases and synthesases, and they do not require water or the electron carrier to break the bonds also. And this enzyme drives the fermentation process in the yeast, turning sugars into ethanol as well as the carbon dioxide by the anaerobic method also, we can say. Next, isomerases includes all the enzymes catalyzing the interconversion of the optical, geometrical, positional isomers also. Isomerases catalyze structural rearrangement within the single molecule. They convert the molecule from one isomer to another without changing the chemical formula. Example, resinases, epimerases, mutases, cis-trans isomerases. So total number of atoms remains identical, only their spatial orientation changes. Phosphoglucose isomerases, glucose 6-phosphate is converting into fructose 6-phosphate, which is a second step in the glycolysis method also. Next, ligases. Enzymes catalyzing the linking together of the two compounds is called as a ligases. Molecular glue also we can call it. Enzymes which catalyze joining of carbon, oxygen, carbon, sulfur, carbon, nitrogen, phosphorus and oxygen. Ligases join two molecules together to form a new chemical bond. Because these reactions are thermodynamically unfavorable, it is always coupled with breaking of a high energy phosphate bond like ATP. Examples are subclasses, synthetases, carboxylases, DNA, RNA, ligases. Require ATP, GTP, similar nucleoside triphosphate as an energy source. Example, pyruvate carboxylase, etc. So, next last one is the translocases. Drive the movement of ions, and they are simply related with the other reactions also. Hydrolases also they will be included. So, thank you for watching. Please do subscribe my channel. Have a nice day.